2 minutes, and then we'll get started in just a minute. Okay, I'm going to go ahead and get started with some of our ordinary background, and then I'm sure we'll have more folks trickle in. So happy Valentine's Day, everyone. We love you all so much. And we're excited to have our meeting today. As a reminder, as always, WastewaterSCAN is a national effort based at Stanford to build this national network of wastewater monitoring sites to monitor diseases through municipal wastewater. So I've got myself, Marlene from Emory, and Allie from Stanford, who are the program directors, leading the call as always, and then all of our partners, Verily, National League of Cities, and all of you. So, say hi, say happy Valentine's Day to each other in the chat, and let us know where you are calling in from. It's so fun to see that we really have a community now. We get to see so many familiar faces on this call every month. Little overview of where we're at with the program. So we still have 147 plants that are sampling across 40 states, representing almost 12% of the US population. This is our map showing all of the sites that are currently in the program. We are currently monitoring among our respiratory diseases, SARS-CoV-2, many influenza targets, influenza A and B, and then also H1, H3, and H5 specific targets, RSV and human metapneumovirus and enterovirus D68, enteropathogens, norovirus, and then our emerging and outbreak pathogens of concern, Candida auris, hepatitis A, Mpox clade 2, and Mpox clade 1b is our most recently added pathogen, which replaced rotavirus on our panel. Reviewing, we have our open access protocols and publications. So we just always want to share with you a reminder that all of these protocols are available, so you can see the methods that we're using to generate all of these measurements and some of the publications that kind of provide the foundation for a lot of the monitoring work. I believe Amanda has also just put links in the chat to where you can find these online, and you can also follow the QR codes from these slides later when we send them out, because I'm going to just keep moving right along. All of our papers and fact sheets are publicly available, so we want to highlight kind of our most recent paper. Allie and her PhD student, Alana, published this paper on respiratory virus season surveillance in the United States over the past couple seasons. So check that out. It's a really interesting paper giving an overview of respiratory disease surveillance through wastewater. Recently accepted paper in collaboration with the California Department of Public Health, led by that team, is this kind of timely, sensitive, and reliable SARS-CoV-2 Omicron variant monitoring in Northern California. So if you remember back when Omicron was circulating, this is a really nice paper about how that data was used. And if you want to see the whole published body of work, you can follow this link, which is also in the chat, to look at all of the publications that have come out of this program. We have a section that's added to the publications page with FAQs, fact sheets, guidance documents. We're regularly putting together these kind of documents when they're needed and requested. And so we have kind of pathogen-specific FAQs, fact sheets, and interpretation and guidance documents that are linked on there. So be sure to check that out, because there's a lot on there that I think is kind of currently useful for things that are currently probably top of mind for a lot of you, including H5, Mpox clade 1b, just conversations on variability in wastewater and how we interpret our measurements. So check those out, because we hope they're quite helpful, and let us know if you have particular requests for other things that you would love to see in this section. We would love to hear that from you. Okay, I'm going to give a quick update on H5 detection in municipal wastewater. I'm sorry. One other thing that I wanted to mention about the program before we move farther is we know we've had some questions, and there's been some uncertainty about funding for scientific research and public health right now as a whole. It's important for you to know and for us to share with you that WastewaterSCAN has not been impacted by any of the funding The funding for WastewaterSCAN has not been impacted. Sample collection, data analysis, updates to the data dashboard will continue for the current phase of the program, and we will continue to submit wastewater data to CDC News. And the agency's website has been updating on schedule, so all of our work is continuing as normal and as planned. We've gotten some questions about that, so we just wanted to make sure that we address that. So I'll talk about H5. So we began our prospective monitoring in mid-May of 2024. As of the 11th, we had 932 positive detections out of over 16,000, almost 17,000 samples. There have been detections at 68 sites across 19 states, and the most recent detections have been in California, Kansas, and New Jersey. So this is all the detections over the entire time period that we have been monitoring H5. You can see the concentrations, a histogram of the concentrations here. So this is looking at what are the concentrations in the frequency of samples that have concentrations falling into each of these categories. So the most common is this kind of relatively low concentration and H5. And then there are a number in this very, very high category. And many of these high H5 concentrations come from sewer sheds that have known dairy processing facilities. So we have seen that a lot of the places where we're seeing that really high concentration are in the areas where we have known dairy input, but there are a lot of sites where we have had H5 detection where that's in relatively lower concentrations that are near the detection limit for the assay. So it's not always that we're seeing high concentrations. I wanted to share this plot that Amanda put together, which shows a really interesting overview of all the positive H5 wastewater detections from the WastewaterSCAN program in this purple at the top here over time, compared to national detections in cattle, poultry, and wild birds. And so you can see these are, in each case, this is the number of positive samples over time, number of positive herds, number of detections in poultry, and number of detections in wild birds. And so it's interesting to look at how we're seeing the pattern of what is observed in wastewater compare to the pattern of what's observed in these animal populations that we know are really important for H5. And so, we're looking forward to talking more to everyone about what we're seeing in these overall national patterns of the H5 detections that we've seen. But one thing that is very interesting to see is the way that, in particular, at a glance, there is this consistent pattern between the wastewater detections and what we're seeing in animals, but particularly between cattle and wastewater samples. We have, among all those FAQs that I pointed out on the website, we have an FAQ for H5. And so Amanda just put the link in the chat, but you can also find that on the website. And there's a bunch of information about how we're approaching testing for H5 and how to interpret and understand those measurements. So please check that out. And again, let us know if you have further questions on H5 or if you have other topics that you'd like us to cover in similar documents. And now I'm going to pass it on to Allie.
Hi, everybody. I'm going to talk about the mpox clade 1b work. Next slide. So the assay we're using detects mpox clade 1b DNA, and it's been previously published by Schul et al. And this is the primer and probe. The data went live on the data.wastewaterscan.org website on January 15th. You can view it the same way you view other pathogens, and you can reference this slide when we send it out so that you can follow it if you're having trouble, but it's under the other category. And then it's shown in default as a heat map. Next slide. We also have a fact sheet, which has some background on mpox clade 1b, how it's different from clade 2, and then how we're interpreting the data and some of the information that supports its scaling. And we're almost done with a manuscript that thoroughly describes everything, which we'll be submitting as a preprint and sharing with you all soon. But a lot of the technical details are also in this fact sheet if you have questions about the assay, how it's why it's different from maybe the assay run by CDC, for example. Next slide. As of now, there have been 4 Mpox clade 1b cases in the United States, 1 in each of the states, which is highlighted there. We have WastewaterSCAN sewer sheds in some of these states, all of these states, but not in the sewer shed where we understand the case was based on news reports. However, that's not the case for California. And interestingly, we did have a positive detection. It's the only positive detection we have had of Mpox clade 1b from the wastewater treatment plant that serves the sewer shed where that individual was reported to be living. So that's very interesting and sort of supports the fact that this assay could work and help identify cases, but only if individuals are located within the sewer shed and that sort of thing. So we're still learning here, and we'll see how this all shakes out. Next slide. Okay, so I want to just talk about some ongoing research to keep you guys updated. This slide has not changed from last time. Just to let you know, we had worked on parvovirus, which we have submitted to a journal, a paper, and I'll go through some of the results. Alessandro Zulli, who's on this call, led that effort. We also are working on Bordetella pertussis, which causes whooping cough. And we're still in the research phase. There's a little diagram there to show you, the steps that we take in developing new assays and scaling them, et cetera. And for the pertussis assay, we're still working on identifying an assay that we think would be good. We've talked about the antimicrobial resistance genes work, which was a cross-sectional study. Some of the data are down here. You don't have to really view those. I'm just showing the occurrence of different ARGs across the United States. That work is still in progress in terms of writing it up and coming up with messages that are of interest, and we'll share that with you as soon as we have it. And then we're also still working on method development phase for metagenomic methods for sequencing viruses, but we're not even close to being able to scale that at this time. Next slide. So just wanted to give you all a really brief update on the parvovirus work. You might remember it in the news last year. It's highly contagious. It causes flu-like symptoms and a slapped cheek appearance, and it has severe complications for pregnant people called hydrops fetalis. And it's usually only diagnosed after people have been contagious for a while because it's syndromic diagnosis. So theoretically, if there was a way to identify parvovirus outbreaks before people were showing up with fifth disease, then that would be useful. Next slide. So in Montgomery County, in Texas, last summer and fall, there were a lot of cases of parvovirus. And so we went back to some biobank samples with the blessing of the wastewater treatment plants in the and the county public health. And we measured parvovirus B19 retrospectively, 2 samples per week, using an established assay. And then Alessandro was able to access the case data from a new research tool called Epic Cosmos, which collects data from electronic medical records and aggregates them in a way that doesn't interfere with patient privacy. So we had aggregated data on parvovirus cases in this area, in this county as well. Next slide. So these are the results from our study. So the top plot here shows the confirmed cases of parvovirus infections in the county. And then the middle plot and the bottom plot show the concentrations of parvovirus B19 in copies per gram from the 2 wastewater treatment plants in WastewaterSCAN in Montgomery County. So visually, you can see the association between the cases and the wastewater concentrations. And there's a significant correlation, of course, between the two. So those are really just the sort of gist of the study. We wrote it up as a dispatch, a short report, and it's currently still in peer review, but it is available as a preprint if you want to look for it. Next slide. Okay, so I'm passing to Amanda now.
Thanks, Allie. Next slide. So I'm going to go over high-level trends of what is happening currently in the wastewater. So just a reminder, we have a category system. I just included a link to where you can find that on our website for the logic. But our categories are based on trends, levels, and frequency of detection. Next slide. So I'm going to go over current categories as of today, as of this morning. So SARS-CoV-2 is in the high category, high concentrations and no trend. SARS-CoV-2 has been detected in 100% of samples over the last 21 days. We're still in an elevated level concentration, even though this map makes it look as if we are going down, we are still in a high category. Influenza A, all sites are currently in seasonal onset, and so we are in this high category due to high concentrations and no trend. Next up, we have influenza B. At the national level, we are not currently in onset, although we are starting to see some regions have more frequent detections. Currently, 39 sites are in seasonal onset. RSV, we are still in the high category due to no trends, but high concentrations. Just for reference, last year's peak was around January of 2024. So we're going to continue to monitor this to see if we are trending downward, if we've hit that max for this season. Human metapneumovirus is still in the low category due to not being in seasonal onset, but the Midwest and the Northeast have hit that threshold for seasonal onset, and we've had a 56% detection rate over the last 21 days. EVD 68 is in the low category due to not being in seasonal onset. There are still 14 sites, though, that are in the high category, and those are sites that are in the Bay Area. So they are in onset, but most sites in the program are no longer in seasonal onset. Neurovirus, we're in the high category. We have a downward trend in high concentrations, but as a reminder, we've seen some very high levels of neurovirus this season, and I'll show a plot in a couple of slides with more detail on this. Mpox clade 1B have all been non-detect in the last 21 days. We've only had one positive detection, as Allie mentioned earlier, since we began prospective monitoring. Mpox clade 2, we've only had 3 positive detections in the last 21 days. C. auris, we've had 26 positive detections in the last 21 days. And Hep A, we've had 123 positive detections in the last 21 days. Next slide. So here's a bit more information on SARS-CoV-2. As I mentioned, it's in the high category due to high concentrations and no trend. But here's a reference plot of the last 24 months, so you can kind of see where we are compared to historic levels we've monitored. We are down slightly in the concentration since January, but we are still in an elevated state. Next slide. Here is the sequencing aggregate for SARS-CoV-2. So if you look at the farthest bar all the way to the right, that is as of February 1st. So KP.3, which is this light blue color, is 32.3%. XCC is 30.8%, and KP.1 is 21.7%. Next. Here is the category map. We are still in the high category for the Midwest and the Northeast, but we've started to see levels come down in the West and the South. And we currently have 54 sites in that high category, 49 in medium, 38 in low, and we do not have recent enough data from 9 sites to calculate categories. Next slide. As I mentioned, I'm going to go over neurovirus briefly. Neurovirus is in the high category due to high concentrations, but a downward trend. We have seen some of the highest levels at the end of 2024 into the beginning of 2025. And as you can see, the plot on the left is the national aggregate of the concentrations. And on the right, you have the CDC positivity rates with data through February 1st. So you can see the patterns mirror each other pretty nicely. Next slide. So here's some historic context for what we're currently seeing in neurovirus category or neurovirus concentrations. We have the national aggregate in black, the Northeast in green, the South in a yellow chartreuse kind of color, the Midwest in red, and the West in blue. And as you can see, we've had some pretty high levels compared to the last 2 years of peaks. Next slide. Here's the category, bright red or bright orange, sorry. Every region is still in seasonal onset with most sites in the high category. Next slide. So that's all we're going to go over for today. We are going to send out the slides that will have more details on all of the pathogens we monitor, and we will send out the recording afterwards. But if you have any questions, as a reminder, you can always email this listserv. It goes to Allie, Marlene, Stephen, and myself. So the 4 of us will be able to pretty quickly answer any questions you have. Next slide. And happy Valentine's Day. Please join us a month from now on March 14th for our next meeting. And we will be sending out the February 2025 newsletter in a couple of weeks. And I'm going to open it up for questions now.